podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hello, and welcome to the Pager podcast episode with Dr. Jim Collins. It's hard to easily describe everything which Jim is involved in, but I think a useful catch-all phrase to use would be that he works on engineering life. In this interview, we get into his work in the early 2000s on designing basic genetic circuits, genes functioning as electrical circuits, which essentially founded the field of synthetic biology. We then move on to talking about antibiotic resistance and how antibiotics might be working in ways which we didn't previously know. This leads us on to engineering bacteria to potentially treat cholera. And finally, we start talking about biotech companies, the relationship between business and biology, what complicates it, and sort of what the benefits are. So without further ado, I give you the Pager podcast episode with Dr. Jim Collins. 3, 2, 1, and we're live. Jim Collins, thank you for joining us on the podcast today.
Well, thanks for having me.
How are you doing?
I am doing great on this beautiful summer afternoon.
It is a beautiful day here, isn't it?
It is.
Yeah, struggled to find my way here, but we're finally off to the races.
Yeah.
So you're interested in synthetic biology and systems biology, is that right?
That's right.
Yeah. Specifically when it comes to synthetic biology, your interests or your research interests are focused around antibiotic resistance and generating these synthetic genetic circuits.
Yeah, so within synthetic biology, we really have two dominant strands of work in our lab currently. One is engineering living cells to serve as living therapeutics and living diagnostics. And second, bringing synthetic biology to cell-free systems, and specifically developing synthetic biology platforms around freeze-dried cell-free settings for a variety of applications, including diagnostics, in-field portable biomolecular manufacturing, and educational kits.
Okay. Now, one thing I noticed when I tried to read through some of the work that your lab has done is, to somebody who comes from like a purely biomedical background, right, which is me, it's pretty hard to decipher all these engineering terms, and the way it's written is, I think, the fact that I'm relatively scientifically literate doesn't necessarily mean that I'm going to understand everything that is in one of these papers. So how can we go about educating biologists about how to think more like engineers, which I suppose synthetic biology is combining these two fields in a way?
Yeah, very much synthetic biology is really at the interface of engineering and biology, and the notion is really using engineering techniques to engineer biology for a variety of new functions and to get after new and different basic science questions. It's interesting to hear your struggles with the engineering paper is coming from a biomedical science background. I remember my early days moving into molecular biology, where I found the original research papers of my colleagues in molecular cell biology to be impenetrable. I viewed them as alphabet soup and could not get past terminology used for different genes and proteins. And it was completely foreign. And I trained myself and interacted with colleagues to learn a sufficient enough amount in order to make my way through. Here, for kind of the next generations, I do think we have an opportunity to train young people like yourself at this interface so that it's not viewed as something foreign, but something that is natural and comfortable at this interdisciplinary, transdisciplinary interface, where you know enough engineering, have a solid enough background of engineering to appreciate the terms and understand their application, but also sufficient background in biology so that you can see where it's relevant and you can see how it can be used to give insights.
And I suspect that schools like MIT and Harvard are actually ahead in terms of, the system is much more modular at undergrad. You can take classes which are much more specific. At least studying medicine, there's no real opportunity. I've never learned about any sort of, there's never been an introduction to engineering and how that can apply to biology and the things we do. Do you know what I mean? Would you agree?
Yeah, so I think our Department of Biological Engineering here at MIT has been a path blazer in largely focusing on molecular level and cellular level questions. That was not dominant in biomedical engineering in the 90s, and the department here was launched in the late 90s. Now it's become much more common to have at least part of the training for biomedical engineers and biological engineers to be at the molecular and cellular level. When I think back to the 90s, biomedical engineering largely stopped at the cellular level and tissue level. But now it's quite natural for 17, 18-year-olds to want to learn how they could engineer proteins or genetic circuits or whole pathways.
Yeah, I suppose it's become so relevant in the last 5 years with all the technology that's coming out, particularly CRISPR and
That's
other
right.
things like
That's
that.
right. Where now we have easier to use
tools,
technologies, ones that are quick, that are accessible, relatively straightforward to program like CRISPR and related gene editing techniques.
What do you think about the role that CS might play in sort of people who are interested?
Yeah,
in
it's an excellent question. To what extent computation,
Mm.
quite broadly, could play. So when I think back to the very early days of what became synthetic biology, our group and Michael Ovitz and Stan Liebler independently were looking to model, design, and build synthetic gene circuits. And in each case, computation served a very big role. And in our case specifically, computation drove our design efforts, and our initial efforts were computational. We didn't have a wet lab, we didn't have money to do the wet lab experiments. And the computation helped guide the experiments we eventually got support for and were eventually able to execute on. I think synthetic biology in its very early days, if you look at the first 2.5, 3 years of papers, they were dominated by computation papers for similar reasons. That the field was launched by a number of misfits from the engineering, computer science, and physics worlds that did not have wet labs or wet lab experience and were looking to see how to contribute, and computation was easy, straightforward. In many ways, the next, say, 15 years or so, computation took a back seat or was not in the car anymore, moving ahead. As we got experimental techniques developed, people were moving and building them without computation. If anything, computation was brought in after the fact, effectively window dressing, prettying up everything. But now we're getting to the stage again as we are building more complicated circuits, as we are in need to find more parts, as we're better appreciating that these circuits are interacting with the whole host, that there's a need for more complex approaches, systems biology approaches. And so we're turning to computation in order to help guide the design, in order to better understand what these circuits are doing.
It's interesting to me that when you refer to sort of the first few years of synthetic biology and the papers that were being put out, I suppose you're also talking about like the Bistable Switch that you were involved in knocking in is not the right word, but engineering in E. coli, is that
Yes.
right? So to me that sounds, for those people who don't know exactly what that system is, that's, am I right in thinking you have essentially two genes, each of which codes for a protein which represses the other gene.
That's right.
And like you change a variable like temperature or something, and this will essentially flip the switch and inhibit the expression of one gene so that that protein that it's producing can no longer inhibit the other gene. Is that right?
That's right. That's right. So it was a genetic toggle switch. So it was a co-repressive system, very much described, or functioning the way you described, where we had them driven by constitutive promoters. So each gene always wanted to be on, but the network was arranged so that each gene was trying to shut off the other gene. And you could, in principle, find the appropriate parameters in the settings so that you could have the system exist as a bistable switch. So it wants to exist in either state A, gene A is on and gene B is off, or state B, gene B is on, gene A is off. And you could flip it between those states by delivering an inducer, be it a change in temperature, or a chemical that would temporarily, or at least while present, shut off the currently active gene, allowing the gene that had been kept up but wanted to be on to come on, and then once it was on at a sufficient level to shut off or keep off the gene that had been on, you could remove your inducer, the change in heat, change in temperature, or the chemical inducer, to now keep that system flipped into the new state, state B or state A.
Now, that I think as a concept, that's not particularly difficult to understand. So it's interesting to me that even at that level, it required a lot of computational sort of analysis. Now, am I right in thinking like sort of the point of these synthetic genetic circuits, defining circuits, I suppose, as genes which grouped together can perform a function for the cell, a useful function. I suppose our interest in them is how can we get, say, prokaryotes to do useful things for us? Is that right?
That's part of the interest.
Okay.
So I think part of the interest certainly is an application, a pragmatic one, and that is, could you use a toggle switch as a form of programmable memory that could record and remember a certain stimulus, or that could be used to keep a certain process on indefinitely and or be able to be toggled between different states. I think it also is useful as a means to better understand different aspects of molecular biology in a very controlled fashion, similar to patch clamp type technology that was used in neuroscience, where you could create small representations or ideal representations or idealized representations of certain aspects of transcription or translation or post-translation, and now studied in a more controlled manner using synthetic biology tools and constructs independent or coupled in certain ways with the host physiology.
Okay, but I suppose what I'm wondering is, do these circuits become much more complicated when you're doing useful things for
You know, it's an interesting question. I think you can do useful things with very simple circuits. And I think the field, which is now nearing 20 years old, has seen some dramatic and interesting applications that have made their way to the market and made their way outside of the lab. But as we get more and more sophisticated, I think the utility increases and the potential application. Now, as one gets more sophisticated and complicated, you also have greater challenges to actually have the circuit function as you like under all the conditions that you like. So it's not in most cases, it's not a trivial extension.
Yeah, so I imagine sort of as they become more like, I suppose an analogy would be like a complicated electrical circuit.
Right.
Is there going to be a need for like computer analysis to actually understand how the sort of the output of these circuits?
I think the computer analysis will be quite helpful, and I think we're seeing the emergence now of coupling of synthetic biology with systems biology. So computational analyses and high throughput measurements of the total host response in order to better understand how these circuits and pathways are functioning within the host. But I think we're also increasingly seeing teams, including our own, turn to deep learning, machine learning, artificial intelligence approaches to improve the design of the circuits and the components so that we can better embrace and better understand and infer design principles that might be useful in biology, in this case synthetic biology.
Okay. Do you think this could help us on to sort of more profound questions about how prokaryotic life maybe even evolved?
You know, yes, I do think that synthetic biology writ large can be and is being used to get after issues around origin of life. Certainly in origin of life, we can at best do a weak existence proof that here is a way that these systems may have arisen or evolved. We no way to ever verify or wind the clock back, but you can identify ways that one may have had a catalytic RNA system that evolved or developed, and there are ways that an encapsulated circuit may have come about, and or a way that a circuit may have increased its complexity through copying and or transposon type activity.
Okay, yeah. Transposons, just for people who don't know, being pieces of genetic information which can, by one way or another, insert themselves into other parts
Exactly.
of the genome. Yeah.
Yes.
Okay.
Yes.
So switching gears a bit, I'd quite like to talk a bit about kind of the work you've done on antibiotics and looking at antibiotic resistance in bacteria. Because obviously, as a medical student, we're taught a lot about kind of how this is going to emerge as, or is emerging as one of sort of...
Yeah, so in this, it's really at the different end of our lab. So we, in synthetic biology, are primarily using engineering approaches to build circuits from the bottom up. We also have a very active component of our lab that uses systems biology. So here, one is using engineering principles to infer the structure of large-scale natural networks and to understand, using the inferred networks, how cells are responding to different stresses. We got excited about using these approaches to study antibiotics about a dozen years ago, and in part, we were motivated to really expand our understanding of how they were acting because the crisis around antibiotic resistance was really just becoming a major public issue at the time, and I thought engineers and systems biologists could make a difference. It's interesting, the next month, September 2019, will mark the 91st anniversary of Alexander Fleming discovering penicillin in London, and you think 91 years is not a very long time, and antibiotics have been marvelous medical miracles, and we've benefited living in the age of antibiotics, and my worry is that the age of antibiotics may be coming to a close.
Yes, and I suppose people, it's useful to put it in the context of how long this has actually been available as a therapeutic tool for us.
Well, you have Fleming's discovery in the late 20s, and really penicillin didn't become available to the early 40s when Howard Florey enabled one to now produce these molecules. So you go back, it's really early 40s, so now when we're late 20s, it's still of order only 80 years that we've had them, but we're running a risk now of not having them. And we were intrigued to see if we could expand our understanding of how they act, and using our engineering principles, we unveiled that downstream of the drug-target interaction, so the general thinking that med students are taught is that the antibiotics will inhibit different essential proteins associated with essential processes, be it DNA replication, protein translation, or cell wall synthesis. And absolutely, that's what they do. Their direct act is that they inhibit or corrupt, interact in some negative way with these key proteins. But then the question is, what happens? How do you get to death? And what we found and uncovered was that there's these interesting stress responses that get triggered, that the cell doesn't like the fact that that process was disrupted and will attempt quite aggressively and actively to remediate. But because the drug is still present, it becomes a futile effort, and the cell will enter and produce different futile cycles that will have significant ATP demands associated with it. So the cell has to satisfy these energy demands, further ramping up metabolism. And that as a result of the ramped up metabolic activity, there are toxic metabolic byproducts that are produced that will damage important molecules like DNA, protein, and various lipids that contribute significantly to the resulting death. And we think this model of the action of antibiotics actually helps to account for many different interesting phenomena, including the challenges of antibiotic tolerance, where a cell will go into a quasi-dormant state and now is not responsive to antibiotic. And why is it not responsive? Well, some have argued it's because the targets themselves are ATP dependent, and I think that's probably partly true. But I think the main reason is that the cell is not in a state to generate these downstream processes. And as a result, the antibiotic is ineffective because the cell isn't trying, it doesn't care.
More specifically, you were talking about the metabolism, the generation of nucleotides is one of the...
So it's one of the features. I think
Okay.
it's a fairly involved and quite extensive process. But yes, nucleotide pools and the generation of nucleotides plays a key role, but it's probably one of probably a dozen different processes. And what's interesting that we uncovered is it's not limited to a single class of antibiotics, that quinolones, like Cipro, fall into this class. Beta-lactams, like penicillin, Fleming's penicillin, falls into this class. Aminoglycosides, like tobramycin, fall into this class. Not all antibiotics are subject to, in a major way, these downstream metabolic activities. Colistin, for example, which appears to act primarily from the outside, punching holes in the bacterium, appears to be only weakly dependent on metabolism.
So essentially what's happening is these cells, they're put under some sort of stress by this antibiotic. Their nucleotide pools are depleted, and they respond by trying to generate more, specifically like adenine, or like you're saying, there are multiple things that can be affected. And that in itself is actually very metabolically expensive.
That's right.
Which damages the cell further.
And it damages the cell further because of the toxic byproducts of the metabolic processes that are turned on. As the cell goes into a massive stress mode, it's as if its house is on fire and it's running around trying to put out the fire, but it ends up doing more damage by trying to put out the fire by kicking. The analogy is breaking down not very poorly here, but its energetic response itself will enhance the killing aspects.
And I suppose there are a couple interesting questions that sort of arise from this discovery. I suppose one thing that's worth pointing out is you mentioned that maybe more reactive oxygen species are generated as a result of this.
That's right.
Which I suppose could also be, in one sense, problematic because you're going to increase the rate of mutation in these bacteria.
Well, that's right. So among the toxic byproducts are reactive species, including reactive oxygen species, if the bug is present in an environment with some oxygen. And we asked that question so that if you had sublethal levels of antibiotics delivered, would you still have some problematic aspects? And by definition, the bug's not going to die because it's sublethal. But we questioned whether you could have increased mutation, and we discovered that in fact that's exactly what happens, that sublethal levels of these antibiotics will still produce this downstream metabolic response, and you'll have reactive oxygen species that will be produced, but they'll be damaging not to a lethal level, but nonetheless at a level that will trigger error-prone DNA repair mechanisms, for example. And we see the mutation rate going up significantly, and thus we discovered that via this mechanism, antibiotics react the active mutagens. So that the use of antibiotics, if the antibiotics don't kill the bug and cure the infection, it could lead to increased rates of resistance.
Yes. Now, I suppose the majority of these mutations are going to be non-functional.
That's
not going
right.
to help the bacteria at all. But what we're worried about are these small numbers of mutations which might confer.
Exactly. So the great majority are neutral or not helpful and disappear. Because in fact, here it's not, our model will be also that because they're active mutagens, they're not simply leading to mutations in the target; they're leading to mutations across the genome. And so you now have these zoo of mutations, most of which will not be selectable, but some of which could be, or will be, and some of which that could be affording resistance in a fascinating and problematic way in the face of also other stresses for the body.
One of the things that I'm wondering about is is there a way we could utilize this? Could we potentiate the effect of antibiotics by adding other compounds which are going to increase the metabolic stress on these bacteria?
Yes, so very good insight. So that's a big part of our program, is figuring out, can we harness these mechanistic insights to improve our antibiotic arsenal? And we've shown, for example, that you can deliver metabolic stimulants, something even simple as metabolites, certain sugars that aren't metabolized by the human host, but still have an impact on the bacterial physiology, and showed that you could significantly boost the activity of antibiotics against these metabolically dormant or quasi-dormant cells. Second is that we showed that if you could, for example, also inhibit the ability of the bacterium to respond to oxidative stress, you can significantly boost the killing efficacy of antibiotics against such bugs.
And so if we do think, if we're able to do this, I suppose that could like extend the lifespan.
Yeah, I think on this we can extend the lifespan of our existing antibiotics. We could potentially pull down antibiotics that had become resistant. Tim Liu and myself showed, for example, that you could engineer bacteriophage that would impact the ability of the bug to respond to DNA damage, and showed that not only could you boost the killing efficacy of existing antibiotics 1,000-fold to 10,000-fold, or 100-fold to 10,000-fold, I think in this case, but we showed you could resensitize resistant strains to antibiotics to which they had grown resistant.
Okay, could you talk to me a bit more about that? Because that sounds fascinating.
Yeah, so there it's interesting, right? So I saw the look of surprise on your face, which your listeners couldn't have seen, and it's very common in that most people think of resistance as a binary event. Yes, no, you're resistant or not, and it's actually not the case. That it's really an analog event. You can be non-resistant, you can be a little bit resistant, you can be moderately resistant, you can be highly resistant, and really anywhere it's a scale. And going back to our comment also on the impact of these metabolic effects, that the metabolic effects will be triggered also at sublethal levels. So they'll also be triggered at different level amounts, super lethal. And what happens with resistance in some cases is that it doesn't completely eliminate the effect of the antibiotic, it just reduces the effect. And so you can envision that now at a clinical concentration, the antibiotic may not be sufficient to be lethal, but it still may be triggering this metabolic response. And so we can shift that threshold with, you have the threshold of, in this case, that's established also by the ability of the bug to correct and protect itself against that. If you now eliminate that, now it's so much more susceptible. And thus, an effect that would not have been lethal and was sublethal now becomes lethal.
Now, how specific can you get these bacteriophages to be? Are you worried about commensal bacteria being affected by these? Or?
The beauty of bacteriophage as potential antibacterial agents or as antibacterial adjuvants is that they have very, very high specificity. So phage are often species specific and in some cases strain specific. So from a narrow spectrum antibacterial development perspective, they offer great, great promise.
Okay. I think I also read something that you had written that I didn't fully understand. So it was to do with sort of potentially using bacteria to, commensal bacteria to mitigate pathogenic bacteria. And I think it might have been something to do with probiotics or gene editing
correct.
Bacteria.
No, it was the former. So we were challenged by the Gates Foundation a few years ago. Could we engineer bacteria to detect and treat cholera? So cholera, as you know, is a nasty gut pathogen that will produce a toxin that will trick cells in our body to get rid of their fluid and outside the extra fluid, get rid of the fluid into the extracellular space. And so if left untreated, you run the risk of dying from dehydration through, let's say, massive diarrhea.
Yeah.
And we took on that challenge. We were able to engineer L-lactis, so a bug that's found in dairy products, and transplanted the quorum sensing system from cholera into L-lactis so it could eavesdrop on cholera and report, change your stool a different color, your poop a different color. But we also found that L-lactis on its own could be a useful anti-cholera agent, and to cut to the answer quickly, showed that L-lactis will reduce the pH in its environment. It'll make the environment more acidic by producing lactate. And cholera hates acidic environments. And so one could use probiotics or bacterial therapy as a means to treat certain infections.
Now, am I right in thinking the way this is working is you are introducing these bacteria who are essentially sensing the presence of only these pathogenic bacteria because of this not operon, sorry, what's the word?
It's actually quorum sensing.
Quorum sensing, yes.
Yeah.
So this means that essentially you don't have to worry about this bacteria, say, making the environment more acidic without the presence of this.
In a next version we would do that. In this case, we did not trigger the production of lactic acid to the detection of cholera. So in this case, they were endogenous, unmodified, and found they were beneficial. One could, however, use or think about using a synthetic biology approach, whether lactose or another scheme, such that the bug would only become active and would only modify the infection site environment once it detected the infection site.
So you could essentially, could you introduce bacteria to somebody's, say, like gut bacteria, which could act as almost like a police? Like they
Yes,
they
you
detect
could.
the
Yes, and
presence of pathogens.
that's a good portion of our lab is focused on attempting to do that.
Okay, interesting. Are you interested in so you looked at cholera? Is there anything else you're interested in?
We're interested in C. difficile, Clostridium difficile, which is a very nasty gut pathogen. We are broadly interested in seeing to what extent we could use such approaches also to refunctionalize the gut microbiome and modify the gut microbiome to go after foodstuff or food allergies, poor food responses like lactose intolerance or gluten intolerance.
It's interesting you mentioned Clostridia, because I know that some of the you've been involved in a lot of biotech startups.
Yeah.
Now I know that at least one of your companies is sort of working on that same question of Clostridia and how to combat that. But you're saying you also work on that with your lab as well.
We do, yeah.
So one thing that interests me, and I spoke to Doug Melton, who you know
Yeah.
about this, is kind of this relationship between the lab and startups for companies.
Yeah.
How do things transition from one to the other?
It's interesting. So we never accept money from companies that we launched. So it's often a one-way transfer, so that on the basis of new developments and discoveries, we, along with our colleagues, will decide that what we have is something that could be useful to launch a company. And we'll do so. And if we continue to work in that area, we often have a friendly relationship with the company. We'll keep them informed of what we're doing, and they'll let us know what they're doing. But it'll be still more one way, that if we have a new development and we protect the IP, if it's protectable, we'll maybe offer it to the company. The benefits, the merits are that I think increasingly academic science wants to have an impact. And an impact we typically have will be through advancing knowledge, training young folks, and then spreading that knowledge through publications. And I see a big shift in the last decade where there's an extra level as well as can those discoveries, the developments, be moved further and into the clinic, into the marketplace. And our lab and many others are getting engaged in startups really to see, can you impact beyond your scientific circle, beyond the scientific community, to now helping improve lives of folks, whether through biomedical advances or environmental advances or material advances. And that's the general upside. The downside, it gets frustrating. You're going to shift something into an external group, and you'll trust a team to try to develop it. And it's very hard to launch successful companies. It's many different aspects of markets being up and down, and very challenging to translate an academic advance into a real commercial product or service.
Yeah. So I suppose one of the most difficult things you're saying is handing off what you've been working on to a group outside of the lab.
And now trusting them to really take it further and develop it. And my skills and experience are not on the commercial side. So when we have brought it to that pre-prototype stage, that's when we then hand it off. But it's not easy to do, to let it go. And say, okay, we now entrust this to you.
But I suppose sort of essential if you ever want what's been done in the lab to really translate into something, like you said, really useful in the real world.
It's essential and important. And that's where it's important also to have them very good commercial partners and startup partners.
Okay, yeah. I'm particularly interested in the different Cas proteins that people are discovering.
Yeah.
And what more is out there, and kind of the different applications of.
Yeah, so there's some really great work being done. Fung Sang is a close collaborator and friend of mine, and Fung has done a very nice job on discovering smaller Cas proteins that lend themselves to biomedical applications. So can you place them on a viral vector that could be used for gene or cell therapy? His team's also discovered interesting enzymes, Cas enzymes, that go after RNA instead of DNA, that also have collateral activity, that they don't simply go after.
Specifically sort of Cas13.
Cas13 or Cas12, which has opened up diagnostic applications. What else is out there? I don't know. It's going to be an exciting time for the next decade or two, given how much interest there is around CRISPR-based systems and how much utility has been demonstrated. I mean, it's been a phenomenal overall advance and really breakthroughs for the field. I suspect there are more and different and interesting things that we'll see coming out.
Let's talk more about this difference between targeting RNA and DNA. I suppose DNA, the changes you're making are much more permanent. What could be the advantages of targeting RNA using a CAS CRISPR system?
From our interest, it's that from a diagnostics end, you can get after a measure of genes that are being expressed. So by targeting an RNA element, you could then be going after pathogens that are in a certain state. DNA would be that they're carrying a certain mutation or certain markers, but it's not that they're in a certain state to go after. From a therapeutic standpoint, it's a similar argument that you can go after the expression state of a given pathological cell, be it a human cell, by targeting its RNA, less permanent as well. That you're really targeting their physiology more so than their genetics.
In the genetics. Okay. Another thing I've sort of read a bit about when it comes to CRISPR is, can we use CRISPR to monitor a cell's transcriptome over time?
Yeah, we can. So there's been nice work done by Tim Liu and George Church separately, both friends of mine, collaborators, where they've developed storage systems or recording systems using CRISPR. And I think that there's a lot of potential there to use it as a means to better understand dynamically what's happening in a cell. By storage, you're still going to have to break open the cell and say, okay, when did it happen? And did it happen? And maybe when did it happen? It'd be nice to see also, could one get after more rapidly turning over reporter systems that you wouldn't need to record, but that you could see unfolding in time as a cell is exposed to a stress or a cell develops, differentiates, to understand what are the different things being turned on or off.
Now, can that be quantitative? Can you tell me at a given point in time how much of a given mRNA there was in the cell? Or is it just
I'm not sure on the recording system if you can. For the systems that we've developed, these paper-based detection monitors, we can quantify the output, so we can tell you. It's always challenging to get after quantification. You got to do certain standard curves and extra steps, but it's becoming more and more feasible to get not simply a qualitative readout of yes, no, to how much.
One of the things I think which is particularly interesting about this is could you use this to better understand disease pathogenesis? Look at a particular cell type in, say, an autoimmune disease, and look at how the T cells behave differently over time as the disease onsets, so to speak.
Yeah, I do think that these techniques broadly, kind of engineering biology techniques, are going to be useful for really expanding our picture of what's happening. I think it's an exciting time to be young, an exciting time I think to be in the space, because over these next few decades, I believe we're going to make tremendous discoveries now on more integrated molecular biology. So instead of just a single protein, single gene, which was the dominant for several decades, it's now how are these collections of genes and proteins interacting, turning on and off to produce the fascinating biology that we see.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.